This Week in Microbiology is brought to you by microbe.tv and is supported by the American Society for Microbiology. To learn more about microbes, visit microbe.tv and asm.org. This is TWiM, This Week in Microbiology, episode 333, recorded on May 29, 2025. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Ann Arbor, Michigan, Michelle Swanson.
Hello. Great to be with you.
333, it doesn't happen very often, every 100 episodes, right? 333, 444. Also joining us from Charleston, South Carolina, Michael Schmidt.
Well, hello, everyone. And last week we had the perfect palindrome on May 20th. It was 5/20/2025. And if you look at it both ways, it's the same date.
Yeah, cool. All these things to keep you engaged.
Entertained.
Also joining us from St. Louis, Missouri, Petra Levin.
It's great to be here. And I don't have much to say other than I guess in the next we'll have to do 111 more episodes before we hit this kind of milestone again.
Well, I'm glad.
You guys have tornadoes there? Is that true?
I'm glad you're okay.
Yeah,
from
we
the
had
tornado.
tornadoes. I was actually flying back from abroad, but my house it scraped. All the shingles are mostly, we're going to need a whole new roof, but we lost a giant oak tree that was on the corner and it fell down and hit our fence. And then it crossed this alleyway and hit my neighbor's garage, knocking down all the utility poles on the way. And we still have the trunk, and a blue jay has decided to build a nest in the trunk. I don't know how long the trunk is there. It's like 10 feet high. It just came up almost like a brownie pan on that stretch between the sidewalk and the curb. It's like a giant chunk of brownie with a 10 foot tall with a giant 70-plus-year-old oak trunk stuck in it. But we were lucky because it was only, I think, an EF2 when it hit us, but it continued on, and another 10 miles north, it took down buildings.
So someone better at probability should do the calculation. My house got hit by lightning. Petra's got hit by a tornado. If I was Michelle and Vincent, I'd be concerned.
About earthquakes?
Yeah.
What's left?
What's left? I mean, floods, fire.
I'm worried about martial law, right?
Martial law, that's true.
There's that.
Yeah.
People coming around just smashing doors and windows.
Yeah.
I'm not sure we have time to do that topic justice.
No, we're going to go into the science.
We're going to do science because science is great and it helps everyone, despite the U.S. seeming to want to break it. It's a wonderful thing, and everyone's lives are better because of science. If you like these programs, by the way, where we talk about science, we give you facts, we give you information, no lies, no misinformation, things that we say usually make sense. We'd love to have your support to continue these podcasts. You can go to microbe.tv/contribute. Microbe TV is the production company for all these podcasts. We do like 9 different podcasts. And it is a nonprofit entity of 501(c), which means that in the U.S., your contributions are federal U.S. tax deductible. Today we have starting with Michael has a snippet for us.
And this is an open access paper. It appeared in April in MBio, and it's entitled, "A Mutualistic Model Bacterium is Lethal to Non-Symbiotic Hosts via the Type 6 Secretion System." And it's by Gaddy, Scepter, Ruck, and Milton. And they are in the Department of Biochemistry and Molecular Biology at the Brody School of Medicine in East Carolina University, and that's in Greenville, North Carolina, as well as the Department of Earth, Marine, and Environmental Sciences at their sister institution, the University of North Carolina in Chapel Hill, as well as the Department of Pharmacology and Toxicology, also at Brody. And it's a fascinating snippet for you today, one appropriate for you to take with you for your beach reading. So we realize it's summer and you don't want anything too heavy. So it's a short paper, not too many complex figures for you to digest, but it's sufficiently provocative for you to tuck it into your beach bag to read and think about while you're harvesting some vitamin D. In short, today's paper is going to challenge our traditional perceptions of the concept of bacterial mutualism and pathogenesis. One of the tenets of the Microbial Literacy Project of ASM back in the 1990s, as the society approached its centennial, was to teach the general public that microbes are not inherently bad. Rather, they are simply looking for a niche where they can thrive without being noticed by a host immune system, predators, or even their nosy neighbors. Case in point is the subject of today's paper. For years, when thinking about bacterial mutualism, one example that immediately would come to mind is that of the gram-negative microbe, Vibrio fischeri, that we associate with the beautiful glowing mutualism it conveys to its partner, the Hawaiian bobtail squid, Euprima scallops. It's the classic example of a bacterium and its host living in perfect harmony, with Vibrio fischeri helping the cute little squid camouflage itself through the phenomenon of bioluminescence. So it turns on the light so that the squid appears to be moonlight to predators rather than today's dinner. And we know that to do its job, the microbe, Vibrio fischeri, employs some pretty sophisticated molecular machinery that we've previously covered on TWiM. Everything from the straightforward molecular switches of quorum sensing all the way to the mechanisms that look suspiciously like what pathogens use, things like the secretion systems and cell surface components that modulate host responses. For more on how this mutualism actually occurs, I dropped into the show notes a paper by one of our former TWiM co-hosts, Margaret McFall-Nye, and her colleague, which is always a fascinating read. And it's a 2021 review, and you can take a look at how Vibrio actually does the mutualism thing. But if you think about it, the big question I want you to think about on the beach, if the microbe has these, if you will, weapons, why doesn't it cause harm to the host when it's living as a mutualist? Or simply put, why doesn't it use its nukes because it's got them? Well, this paper throws a fascinating wrench into the narrative of mutualism, and Gaddy and their colleagues, they took Vibrio fischeri out of its comfy squid home and exposed it to other aquatic macroorganisms that it might encounter in the wild, specifically zebrafish embryos, and then another larval form of a shrimp, Artemia napoli, that we affectionately refer to as brine shrimp. And lo and behold, they found that Vibrio fischeri could indeed go from being a mutualist to becoming a formidable pathogens for those new now hosts. They approach this from the vantage point that the type 6 secretion system, and this is a specialized export system used by gram-negatives to move a protein effector molecule manufactured by the bacterium from the interior of the bacterial cell across its two membranes, remember the unit membrane and then the outer membrane of the gram-negatives, into the adjacent, in this case, eukaryotic target cell. And as an aside, it's estimated that at least 25% of all pathogens and non-pathogenic proteobacter, and so this encompasses a large swath of gram-negative microbes, encode a type 6 secretion system, with the genes generally residing within the bacterial chromosome. Aside from moving an effector molecule into another cell, type 6 secretion systems play a role in the acquisition of essential metals, such as manganese and iron, which, of course, impact fitness on the microbe from the surrounding environments. This ability allows the bacterium to then outcompete rivals for these essential nutrients while fostering cooperation with related bacterial cells, because when the type 6 is throwing out, if you will, a siderophore, it's now a public good, so it's benefiting those nosy neighbors that we introduced you to. This suggests that type 6 secretion systems play a critical role in maintaining microbial community stability by balancing such cooperation and, of course, competition. Now, recall that the majority of Vibrio species possess 2 chromosomes. This is an uncommon feature for bacteria, as most only have 1 chromosome. We have already offered that type 6 is an important virulence factor for many pathogens. But since Vibrio has 2 chromosomes, they also have 2 type 6 secretion systems. So first question is, which one matters? The one on chromosome 1 or the one on chromosome 2? And the authors will actually answer that question for us. So they approach the challenge of which one matters by testing the conserved type 6 secretion system from Vibrio fischeri. Specifically, it's located on the chromosome 1 for its potential role in pathogenicity in these 2 other aquatic species. Their hypothesis was that the type 6 secretion system on chromosome 1 was involved in the eukaryotic interaction. Up until this point, it was unknown what the type 6 secretion system on chromosome 1 actually did in Vibrio. They knew that type 2 was important for the mutualism behavior, and that has been worked out in many other fine papers. And so they tested it in the zebrafish and the brine shrimp. In the first figure, they give us the first bit of data that where they observed in a dose-dependent mortality experiment in both the zebrafish embryos and the brine shrimp, that mortality increased as a function of how long the larval forms of these macrobiotic organisms were exposed to the microbe in solution. And they learned that it wasn't a consequence of a secreted toxin. They did that by filtering the media. Rather, they showed it was actually by the live Vibrio fischeri cells were required for conferring this lethality. The bacteria were actually associating with the host tissues, suggesting a direct interaction. And recall that type 6 secretion is really nothing more than a molecular harpoon. It's stabbing the host cell to inject itself into it. So they have to, it's not a great stretch to think that these microbes need to come in contact with the host. But even more intriguing, the zebrafish that survived exposure showed significant developmental defects that you can witness in the figures. And you can actually see, we're talking stunted growth, necrotic tissue, and even abnormal tail curvatures in the cute little zebrafish.
I know, poor zebrafish in G. And I think this is worth looking at carefully because it's very helpful because sometimes in these pathogenesis papers, they show you thin sections, for example. And here they're pointing out exactly like the blood pooling and the tail. And it's actually really instructive to look at because most of us don't look at zebrafish embryos. So I commend them on having these really helpful figures.
And they talk about how the protein that's being injected potentially, we don't know which one matters because they didn't identify that, but it's beginning to slow down the heart and begin to clot blood and literally stop circulation. So this is clearly not a beneficial interaction. We're not talking mutualism. This is God-fearing pathogenesis, folks. They did confirm zebrafish survival can, of course, be recovered by the control. Namely, you treat the Vibrio fischeri with streptomycin, or you heat kill the microbes prior to exposing the zebrafish or the brine shrimp, and as expected, the cells aren't harmed. And finally, to test whether mortality was caused by the accumulation of these extracellular compounds, they took the E3 media that the Vibrio were incubated in filter sterilized it before the embryo immersion. And again, survival rates were similar to the control. So we've established that zebrafish mortality was dependent upon viable fisheri interacting with embryos. Now, because of this dose dependency in spent media experiments, they then predicted that Vibrio need to be in direct contact with the embryos to exert their lethal effects. So next up is an association experiment where they measure colony forming units to get us to the experiments in panel C of their first figure. And here they took washed and homogenized embryos with the chorion removed. And again, they're using millions of bacteria in order to effectively see this. And they're showing that the microbe doesn't need to be localized to the embryo surface in order to do its dirty deed. So then they posit the following question. What's the mechanism behind this Jekyll and Hyde behavior of Vibrio fischeri, where in one instance with the bobtail squid, type 6 secretion is beneficial for the light organ, but in these other animal models, it's lethal. So what virulence factors are driving the lethality? And here's where they get to answering what type 6 secretion system is essential for this behaviorism. And so here they point their finger at type 6 secretion system from the first chromosome. And if you think about it, when we look at the fact that it's got two, and we know that the type 6 secretion system on the second chromosome functions as an actual antibacterial weapon and is used by the microbe to establish its monocolonized crypts in the bobtail squid, especially for its like organs. That's why it's effectively in a xenic culture of the Vibrio fischeri in the light organ of the bobtail squid. Type 6 secretion system from that first chromosome is conserved in all of the isolates that they looked at. But again, it has an unknown function. And the type 6 secretion locus from the first chromosome is not essential for squid colonization. So to determine the extent to which the Vibrio fischeri lethality is conserved across strains and the role of that type 6 secretion system from the first chromosome, they use 3 symbiotic fischeri strains, ES114, 213, and PP3, and they contained a deletion of the type 6 secretion structural gene, ICMF, which encodes essential structural protein for the type 6 secretion system. And they compared it to the wild type genes of these 3 strains in both the brine shrimp as well as the zebrafish studies. And they did survival studies. And these studies provide direct evidence supporting their hypothesis that where they found that when they deleted the ICMF1 gene, again, a structural component of the type 6 secretion machine. So we don't know what the lethal bit going out of the apparatus is doing, but we know that in 3 different fischeri strains, what they found was pretty interesting, albeit a bit nuanced. The role of type 6 secretion in lethality was both host and strain specific. For instance, deleting the ICMF1 gene in strain 213 significantly increased survival. That makes sense because in the brine shrimp, it needs that accessory protein that's coming from the bacterium to kill it. And so if you delete the component of the secretion apparatus that's responsible for sending it over the side, you would expect survival to increase. However, for ES114, type 6 secretion seemed dispensable. It wasn't needed for Napoli lethality or the brine shrimp. Now, the picture changed slightly for the zebrafish embryos, while the ES213 deletion mutants still cause lethality similar to its wild type parents, again, suggesting type 6 wasn't essential for this strain's effect on the zebrafish. ICF-F1 mutants of 114 and PP3 showed higher survival rates. So we got a conundrum here. So this strongly suggests that under these experimental conditions, type 6 secretion is indeed involved somehow in Vibrio fischeri pathogenesis against its eukaryotic host. This isn't unheard of for other Vibrio species like Vibrio coralliticus. They have different type 6 secretion systems with distinct roles, one for bacterial competition, one for targeting eukaryotic cells. So there still may be some other secretion system that hasn't yet been worked out in Vibrio fischeri, or somehow the effector molecule is somehow getting in to these cells. And that's where they leave us. What does this all mean? It means Vibrio fischeri is not just a microbe with dual capabilities. It's able to both have beneficial and pathogenic properties, but you need not necessarily always need this type 6 secretion apparatus from this first chromosome. So this goes to show you that pathogenesis is extremely nuanced and there's a role for everything.
Michael, in nature, are zebrafish or Nauplii infected with this microbe?
I doubt it, because I don't think zebrafish are in the same water column as the bobtail squid in Hawaii, but Margaret could probably address that better than I.
It's not surprising that if you end up in the wrong host, you cause disease. That happens all the time with viruses.
Oh, yes.
Right.
Viruses are in one host, they do nothing, and then they get into another host, and boom, you have disease. Now, in the one host, we don't know if they're mutualistic or not. It could be, but we don't know. So that's the unique thing here, is that it's a mutualist that then becomes nasty. But yeah, viruses do this.
So this is a bacterium that wants to become a virus?
Well, I mean, what I was struck by is we use actually Xenorhabdis and related species when we teach mutualism and virulence, and kind of they're two sides of the same coin, because they both involve colonization and, receptor mediated responses and all this stuff. And so I always use this Vibrio fischeri as mutualism, and now it has this pathogenesis side to it. I would be curious though, because in the Xenorhabdis, right, it's the mutualist with the nematode, and then it teams up literally with the nematode to kill the insect larvae. Here, it just seems kind of opportunistic.
And I think that's what I would say. This is truly an opportunistic behavior where the microbe is out to get food.
Yeah, it is also, though. It's just a good reminder, like we teach the students, mutualism and pathogenesis are two sides of the same coin.
It's like if a tree falls in the wood, it doesn't make any noise. It's the same idea here. If this never infects these hosts in nature, what does it mean?
Yeah.
Yeah, I mean, I wonder what it does infect. Like if you pull, because the squid lives in, I don't actually know where zebrafish come from, or if they're fresh or saltwater fish.
I don't either.
Which is embarrassing. I'm sorry, zebrafish people.
We're going to get letters for sure.
We're going to get letters. Please send us letters. We will call you guys out if you send us letters. But I mean, the fact is Vibrio in general.
They're freshwater.
They're freshwater. Okay, so these guys probably never see each other, so the zebrafish. But this is actually, I think, pretty interesting, because Vibrio, right, cholera is not, it's in lakes and streams, especially, it's famous for being in
Bangladesh
European
rivers,
streams, and then brackish water. But it really causes disease in part because it is the ones, especially like El Tor, have a phage that makes them hypervirulent. So it's kind of like, what is going on with all these different bacteria? So, and what is making them virulent or not? And like you said, Vincent, with viruses, sometimes, they're mutualists or commensals, and sometimes they're cause disease. And
it's even more complicated because it's got two chromosomes.
Many virulence factors need to have a particular receptor in order to mediate their effect. And so their virulence is going to depend not on what's in their genome, but also on who they encounter and what they encode.
Right. So that's what I also didn't. That was my other question. Sorry, I'm just kibitzing here. But with the type 6, do they have a receptor on the host cell, or do they just kind of any host cell.
Well, they talked a little bit about this, about how the microbe can go through, if you will, the walls that we anticipate in this developing form of the zebrafish, and how the microbe can make direct contact with the zebrafish structure. I guess, for the lack of my anatomical knowledge about zebrafish and what they need to be called, but it's sort of they're coming in close proximity to them, where they need to inject that, the harpoon metaphor holds. They're effectively injecting something into the host.
And in the harpoon metaphor, does the harpoon itself cause damage?
Yeah, that's the other thing. So there's many questions from which they can continue on this journey. And I thought this would be nice to think about as you're soaking up that needed vitamin D.
So the thing that's interesting here is that you can find a you got type 6 secretion system. That's a very specific mechanism, right? You can't do that with viruses. So SARS-CoV-2 jumps into people, it makes them sick. It's probably just a matter of immunity, population immunity, right? And maybe in its natural host, the bat, they're all immune, and that's it. But
Right.
you can't point to any specific gene product that is responsible for this.
Yeah. And Arturo Casadevall has written essays about this.
Yep.
And I'll drop that essay into the show notes.
Thank you, Michael. Okay, now we will move to tuberculosis with Michelle.
Yes, the paper is a BCG kill switch strain protects against Mycobacterium tuberculosis in mice and non-human primates with improved safety and immunogenicity. It was published in the February issue of Nature Microbiology, and it is a collaboration between 3 labs, Dirk Schnappner and Sabine Ehrt at Weill Cornell Medicine and Joanne Flynn at University of Pittsburgh. And the 3 co-authors are Alexander Smith, Hongwei Su, Joshua Wallach, and their other co-authors are Lou, Mayello, Borish, Winchell, Simonson, Lynn, Rogers, Fillmore, Sakal, Lynn, Vinette, and it is a exciting development in the treatment and prevention of Mycobacterium tuberculosis. So this bug, Mycobacterium tuberculosis, is the #1 cause of infectious disease, and that is thanks to the triumph of the SARS-CoV-2 vaccine, which that virus had overtaken MTB, but MTB is back now. Each year, it causes more than 10 million cases of infection worldwide, especially in Africa, Southeast Asia. Here in the US, it causes about 10,000 cases annually, and most of those are from people who were born abroad. The incidence is actually 20 times higher in people born elsewhere. Humans are its reservoir. We spread it by coughing. If you inhale it and it gets into your lung, we have white blood cells that patrol the lung and eat and digest particles. But not TB. TB, once eaten by a macrophage, can actually set up replication niche there and replicate. But if the person has got a robust immune response, they will trigger a local defense and actually wall off the infected cells and the replicating bacteria within stable structures called granulomas. And these are really great systems for containing the infection. TB can persist there harmlessly in a human lung for years, for decades. It's actually estimated that 1/3 of the world's population actually harbors latent TB in these granulomas in their lung. Problems arise if the immune system is weakened by aging or immunosuppressive therapy or HIV infection that's not being treated. Now, as the immune system wanes, TB can wake up and start replicating and spread throughout the body. Also, the other problem is that newborns don't yet have a robust cell-mediated immune response, and they're vulnerable to systemic disease. So more than 100 years ago, a French physician, Calmette, and a veterinarian, Guerin, developed a vaccine. And they did this by isolating Mycobacterium bovis from cows, passengered it more than 200 times on a media made of potato slices, bile, and glycerin. And in the process, this bovis strain accumulated a chromosomal deletion and other mutations that neutralized its capacity to cause disease. So this strain is famously known as Bacillus Guerin, and it is now given to newborns. It was initially, to test it, was given to newborns of families who had TB, and it was found that the babies were protected from severe disease. So for more than 100 years, this BCG strain or derivations of it have been used as a vaccine, and it's estimated that 90% of the world's children do receive the BCG vaccine. It's our only option. We actually don't use it here in the US because the low prevalence of BCG, and we also want to preserve the use of a classic tests of reactivity to PPD. Anyway,
But what we do use it for in the US is to treat bladder cancer. It's amazing.
that's
is.
right. Yeah, it triggers a robust immune response.
Yeah.
That's exciting.
Unfortunately, 5 to 8% of the patients could die of it. So,
Yeah,
it's a trade-off. Like,
BCG is saving a lot of people, but it also has some risks, and that's really what motivated this study. So because it's a live attenuated vaccine, which means it's a replicating bacterium that is put in people's body, if they've got weakened immune system, they can be at risk. And again, in infants, it can cause meningitis. So why haven't we just developed a heat-inactivated or subunit vaccine? The problem is that those stimulate antibodies predominantly, which of course can't get at the MTB because they're replicating inside white blood cells. So instead, we need to simulate T cells that can recognize and kill infected cells and also release cytokines to activate macrophages to contain the infection and build these granulomas. So that means you've got to have get the antigen inside cells. So that's why they're using the live attenuated BCG. But recognizing that this vaccine still poses some risk, this team have harnessed the awesome power of bacteriophage and bacterial genetics and designed a strain of BCG that can still infect and replicate in white blood cells, but they've also engineered it to carry a kill switch that the scientists can turn on or off at will. So that is the key for this new development. So to make this strain with the kill switch, they leveraged a couple toxins encoded by bacteriophage that are known to infect and lyse mycobacteria, but not all the beneficial microbes that we now appreciate we rely on for health. So it would be specific for mycobacteria. So they did this by cloning genes for particular lysins that can, as the name implies, lyse open mycobacterium. But they've engineered it so that it is expressed behind a composite promoter that can be turned on or off in the presence of particular antibiotics, derivations of or derivatives of tetracycline. So this they call, the one I'm going to focus on today is their BCG TET-off DL, and that stands for the two different lysins that this promoter drives expression of. So I won't get into the molecular details, but it's a really clever on-off switch for turning on lysins that will bust open mycobacterium and kill them. So first they wanted to see if this engineered strain still grows normally in broth, and they did that experiment shown in figure 1C, and they showed that if they add tetracycline, they see a 3-log drop in CFU, and that's because the tetracycline will then turn on expression of these lysins, the toxins that they put into the strain. And they could tell that the bacteria actually lysed because they saw a sharp decrease in the density of the bacterial culture. So this is a standard method. We measure optical density of the culture. So far, so good. The strain is being killed when they add the tetracycline. They then wanted to test if the same could be observed in mice. So they did an IV injection of mice with their BCG TET-off DL strain, and they gave the mice chow that contained tetracycline, which turns on this promoter. And they found that the strain persisted in both lungs and spleens, but when they removed the supplement, they de-repressed the promoter, and now they got lysis and killing of the vaccine strain, this BCG TET-off DL. So that was consistent with their design. So now they've seen it both in broth and in healthy mice, but they wanted to do a more rigorous test of just how effective their off switch is for clearing this vaccine strain. So now they did the same experiment with mice that lack B cells and T cells. So these are called SCID mice. So they're immunosuppressed. Fortunately, they got the same result. When they activated expression by withdrawing tetracycline from the mouse diet, they saw that the CFU of their vaccine strain steadily declined in the lungs of the mice and also the spleen, despite their being immunocompromised mice. So they were really encouraged by this safety, and we're now ready to look in more detail about the ability of this vaccine strain to elicit a robust immune response. So here they did an IV vaccination of mice and then looked 3 weeks, 8 weeks, and 12 weeks later at, for example, the populations of white blood cells in the lungs, and they compared it directly to the parent classic BCG vaccine strain. And fortunately, they saw in figures 3A to C that similar populations of our good guys, the CD4 and CD8 T cells, were generated by this TET-off strain, the kill switch strain. Not only that, but now that when they exposed those mice that were vaccinated with their TET-off strain with infectious TB, they found that the BCG vaccinated mice and the BCG TET-off kill strain mice had a tenfold reduction of MTB in their lungs. And also in their spleens, they found that the BCG classic vaccine reduced the MTB CFU by about 1,000-fold, and their TET-off, more safe strain reduced the burden also, but only about 30-fold. And they reasoned that that probably reflected that they had cleared the BCG TET-off DL strain with their kill switch. So the mice were exposed to it less time. So perhaps that's why there was a less robust response. So they're really encouraged. They've found that they can cause this vaccine strain to basically commit suicide when they change the exposure to the inducer, tetracycline. And they're finding it elicited an immune response that was protective in mice, even mice that were immunocompromised. So they were confident enough then, both in their efficacy data and also the safety profile, that they decided to move their experiments into a non-human primate model. And for this, they were fortunate to collaborate with Joanne Flynn at University of Pittsburgh, who is an immunologist with extensive experience using the macaques and to study TB pathogenesis. So their first order of business, of course, was to do no harm. They wanted to learn whether their BCG TET-off DL vaccine strain would be cleared or would it persist after they inoculated the macaques. So they did the IV inoculation and fed the macaques or treated them daily with tetracycline, which again is how they control the expression of their the license that will cause the kill switch. And then after 2 weeks, they removed the inducer of the lytic proteins and they quantified the burden of the vaccine strain, as well as looking at the cellular immune response in the bronchial lavage fluid, so in the lung fluid. And they also did a careful evaluation of tissue histology, all in the macaques. So fortunately, they saw a similar robust immune response, similar profiles of particular classes of CD4 and CD8 memory cells, cytokine producing T cells, et cetera, in the macaques, whether they were inoculated with the classic BCG or their BCG TET-off DL kill strain. They also saw that the histology of the lung, spleen, and lymph node was unremarkable, so no untoward pathological effects there. And by 8 weeks, they found that the burden of the vaccine CFU had declined 4 logs, and they didn't recover any viable bacteria from the blood, the liver, the kidney, or the sternum. So based on these and other experiments, they were confident that they were doing no harm by using this vaccine strain in the macaques. So now they were ready to test directly whether this vaccine strain would actually protect the non-human primates from an MTB infection. So again, they went to their macaque model and they inoculated with BCG or their kill switch strain, or neither, and then treated the animals for 2 weeks with tetracycline to turn on the lysine expression and trigger the lysis, and then waited 20 more weeks before instilling MTB in the bronchial passages of the vaccinated and the control macaques. And then over the next 12 weeks, they profiled the immune cells in the bronchial fluid, and they also monitored the lung and lymph nodes using PET and CT imaging. And they also did painstaking quantification of the burden of MTB, assessment of the lung and spleen pathology, and also analysis of the T cell populations in the bronchial lavage fluid. So they generated really a treasure trove of data for the mycobacterial field. And I'm sure this is out of their respect for using these macaques for this important biomedical research. So by multiple measures, the BCG TET-off DL vaccine strain protected the macaques from MTB. So for example, 75% of the animals vaccinated with the kill switch BCG had sterile tissues, whereas with the classic BCG vaccinated tissues, only 25% were sterile, and that's shown in Figure 5F. Again, they did really detailed immune profiling. They found in the lungs, for example, that the populations of CD4 and CD8 T cells that were making pore-forming toxins that can kill infected macrophages and generate cytokines to activate macrophages to block replication were similar, whether they vaccinated with the classical BCG or their kill switch BCG strains. They also, because they were able to control at what point after inoculation they triggered the kill program, they were able to determine that it only takes a couple weeks of exposure to the live attenuated BCG to stimulate a broad, robust, cell-mediated immune response that protects the animals against MTB. So that was really key. In fact, they found that the program lysis of BCG actually generated a larger population of CD4 positive T cells and even more interferon gamma and TNF in the lungs. And these are two cytokines that are critical for mounting a robust defense against mycobacterium. And they reasoned that probably the ability of their kill strain to generate a more robust response was likely due to the fact that when they triggered lysis, they got a release of just a whole gamish of mycobacterial cell fragments, which are well known to stimulate host defense pathways and inflammation. So that probably contributed to the robust broad immune response. They also wanted to do another really important measure of safety, and that is how genetically stable is their kill switch vaccine strain. In particular, during the 8 months that they followed these animals after vaccination, do any mutants arise that can escape killing by the license? So, for example, did spontaneous mutations arise that made the cells, bacterial cells, immune to the license?
We've learned our lesson from polio.
Yeah.
Yeah. And if such escape mutants did arise, they would be a risk to people that have weakened immune systems or to infants. Fortunately, we got more good news. They calculated a rate that was incredibly low. 2 times 10 to the minus ninth mutants per cell division is what they deduced was the mutation rate. So this is within what they can calculate would be a safe range. This kind of reinforced the design where they decided not to deploy just one lysine to kill the vaccine strain, but rather 2 that work by different mechanisms. So that would decrease the probability that any one bacterial cell could randomly acquire spontaneous mutants and escape their kill switch design.
And for the engineers listening to the podcast, the mutation rate of 1 every 2 billion is well within the safety margin of the 6 sigma safety factor that most engineers use when designing airplanes, bridges, tunnels. They look for a 6 sigma error rate. And the fact that this is more than 3 orders of magnitude greater, they've really over-engineered this reversion rate so that it's truly, truly safe.
Not only that, but two of the authors here, Dirk Schnappinger and Sabine Ehrt, actually they have a pending patent application for this particular kill switch BCG strain, but they've also collaborated with two other thought leaders in the TB field, and that's Sarah Fortune and Eric Rubin at Harvard Medical School, to use similar strategies to design a triple switch kill strain of not BCG, but of MTB itself. And they published this in a companion paper in the same issue of Nature Microbiology. And they basically demonstrated that the kill switch works. Yes, they can kill off their vaccine strain by treatment, controlling the treatment with their inducers, which again are antibiotics. And it also, they demonstrated that it was genetically stable, even more stable, Michael, than the 2 lysin model. And they found that the strain was killed not only in healthy immunocompetent mice, but also in mice that have an immune compromised mutations. So they're encouraged that this MTB strain could also be taken in further studies to perhaps design an even better vaccine. And along those lines, I want to point out that these groups are part of a global effort to combat TB globally. So there is a consortium organized by the World Health Organization that more than 10 years ago adopted an NTB strategy. And they set really specific, ambitious goals for multiple facets of a global effort to diagnose, treat, and eliminate TB. It's now called the Stop TB Partnership. There are more than 2,000 partners across the globe, and currently there are 15 different vaccine candidates in clinical trials and 29 different drugs in clinical trials. So I am proud that the community of TB researchers and public health workers are working together across the globe to come up with multiple strategies to protect people from TB infection, which again, number one cause of infectious disease across the globe today.
Michelle, what do you think about intravenous immunization? I mean, it's hard, right?
It is hard, but it's a good place to start. I mean, certainly with their precious macaque animals, I can see why they would do the IV injection. But
we've done it with
ultimately,
malaria.
Mm.
That's the way one of the malaria vaccines works, through IV.
I was thinking this is like a great example of, first of all, lots of people working on a very hard problem that's been with us for a very long time, because BCG is not that effective. That's my understanding also why the U.S. has decided not to use it. As vaccines go, it's not like a 95% like a COVID
Yeah, it's
vaccine.
very good in the pediatric population, people that are especially vulnerable.
But it's not, for adults, it's not as, but that they're really, kind of like the HIV vaccines, like they're really trying different things. And I kind of see this as like the technology might not be initially very easy for delivery. And especially in the situations you're going to need to deliver it, for big populations. But I think it's a great start. And this, I kind of thinking outside the box, you really need to have a whole bunch of different groups trying to tackle the same problem in different ways. So I kind of love it as from a technology standpoint, it's really interesting. And also this is very clever.
Speaking of needing multiple people contributing to this effort, tell us about our 3 first authors.
The author that Joshua Wallach is one of the 3 co-first authors on this, and he's actually been in the lab since he was an undergraduate at NYU. And as an undergraduate, he planned to go to med school, but then he came to the Schnappinger lab, and he was wanting to get real world experience and kind of bolster his application. for med school, which a lot of students do, and I think is great. It's great if our MDs really have some experience of what goes on at the bench. But he really fell in love with the lab, and he's been basically in the lab ever since then. And he's had different titles: part-time technician, and he's now a senior research scientist. And he does a lot of the screening of novel drug compounds, generating, testing new mutants in MTB and BCG. And he's always doing multiple things at a time. And he's also here to give guidance. So it's amazing to have somebody in the lab like this who can mentor inside your lab in addition to you as a PI. It's great for the people in the lab to have this long-term knowledge. And then he has been working on this project since 2014, since he has a 7-year-old daughter, so much longer than she's been on Earth. And he said that really his fondest memories are when they finally identified a strain that demonstrated the level of safety, because obviously with a disease like TB and a vaccine that's using whole cells, right? It's not like a protein or like part of spike, like in the RNA, you're using whole cells. There's a huge level of safety. And so that they finally identified a strain that did that. That was a really meaningful and important day in lab. And that allowed them to move on to the non-human primates and collaborate with the Flynn lab. So yeah, I'm very impressed. And this is a somewhat different kind of route to being a first author than we normally have. And I really appreciate that, he was inspired and has been working on this problem for a long time.
I'm sure he's worth his weight in gold.
Oh,
to be able to train the new people that come in, and obviously very good at the technical components.
Yes.
We have two other co-first authors. One is Alex Smith, who is at the moment working at University College London on streptococcus, respiratory tract infection, correlates of protection. He says he has a passion for vaccine research. He worked at the University of Pittsburgh, of course, on this paper, and got his PhD at the University of Surrey. He's originally from London, and actually his love of microbiology developed in high school when a passionate biology teacher introduced him to the world of microbes. I know that experience. I had a great biology teacher in high school, too. He enjoys in his spare time bird watching and photography, and also is a Pittsburgh Pirates fan, unfortunately. I guess they don't win. That's why the unfortunate.
Not since the 70s.
He likes to point out the important collaborations of the team at Pitt and Weill Cornell. It's a theme today. He says, the veterinary staff, technicians, and imaging people makes the non-human primate studies possible. Collaboration and teamwork is key. And he wants to thank Joe Ann Flynn, his mentor, for taking a chance on a guy with minimal immunology experience. And he said, if I were to give one piece of advice, if you're contemplating making a move to a new city or a country, do it. When he was interviewing with the job in Pittsburgh, COVID restrictions were in effect. He'd never been to the U.S. before, but the decision to move was one of the best decisions in his life. It was an adventure that led to fantastic science, introduced awesome places and incredible people. So even though you have to uproot your family, sometimes it's worth it. And then the other co-author is first author Hongwei Su. Has been working on MTB research since his PhD work, which he got in 2017 from Tsinghua University in China with Professor Babak Yavid, focused on regulation of translational fidelity. In 2014, he was a visiting researcher in Bhavesh Khanna's lab at the University of the Witwatersrand in South Africa. And for a postdoc, he joined the lab of Professor Sabine Ert and Dirk Schnappinger at Weill Cornell, where he worked on the latent TB model and he calls the self-destructing BCG vaccine. He's very grateful for the mentorship from Sabine as Dirk, as well as the support he got from all the colleagues in the lab. So again, thanking other people. So he wants to thank Dr. Alex and other members of the Flynn lab who worked on the non-human primate study. Great example of collaborative research across multiple labs. He's currently a principal investigator leading a small lab at Zhejiang University in China, focusing on mycobacteria single-cell heterogeneity and the mechanisms of drug tolerance. So science is an international endeavor, folks.
And if you think about it, if you look at this BCG vaccine candidate, and since we're recording this the week that Mission Impossible has come out, this is truly, should this strain accept the mission of being a vaccine, it will self-destruct once it's performed its task.
What do you mean it's just coming out? They made a movie or something?
Yeah, they're the newest Mission Impossible where Tom Cruise saves the world again.
But I thought it was a TV show years ago, right?
It was, but they've had like 9 movies.
I don't know anything about that.
You got to go to the show, Vincent.
The movies are amazing, and Tom Cruise does all his own stunts, which is also, inspirational as somebody who's in middle age.
I heard that they just did a remake of Day of the Jackal, which I saw maybe 30, 40 years ago, and I thought was fabulous. So I don't want to see a remake because the original is what I loved, right?
Well, you remember from the original TV show when the tape recorder self-destructs. And that's
I remember that.
And that's effectively what the BCG is doing once you get the IV construct. It's self-destructing
right?
after it gives you all the correlates of immunity.
That's right.
It's doing it in a Mission Impossible. Yeah.
BCG vaccine has been in use for 100 years.
Yeah. It really is.
But I'm optimistic. I'm feeling good about their efforts and people worldwide. I think we're going to make headway.
A third of the world's population has TB. 1/3 of planet Earth.
Yeah.
That's a lot of people.
There's not much in the U.S., right?
No.
Right.
There are some, right? But is it growing or it's just stable?
It's fairly stable. It's the incidence, as I said, in people born elsewhere is 20 times higher than people born in the U.S. So it's travelers.
It's because of good public health programs where they aggressively go out and identify it and then put the people on direct observed therapy where they literally watch you take your drugs in order to control.
Public health is out the window in the U.S.
I'm afraid, Vincent. I am afraid.
So maybe we're going to see a resurgence of TB, among other things, right?
We're going to get through this. Just a question of how much time.
Yeah. I'm glad you're optimistic, Michelle.
This is a good example, though, I think, of the surveillance with the the test, the PPD test, because if you go to Europe and the only way they have to test because everybody's vaccinated with BCG is to get a chest X-ray.
Yep.
Or the more expensive quantiferon test. The quantiferon
it's also heartening that there is global collaboration still.
Yeah.
Oh, 100%.
Yeah. Even though
global collaboration. We're all for it.
The U.S. has pulled out of WHO, is my understanding.
I hear they're going to make their own WHO. The U.S. and Argentina are going to make their own WHO. Can you imagine? Ridiculous. Reinvent the wheel. All right. That is TWiM 333. Another 100 episodes before you get to 444. You can find the show notes at microbe.tv/TWiM if you like our work. Please consider supporting us, microbe.tv/contribute. And should you have any questions or comments, TWiM, TWiM at microbe.tv. Michelle Swanson is at the University of Michigan. Thank you, Michelle.
Thank you.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
And Petra Levins at Washington University. Thank you, Petra.
Thank you.
I'm Vincent Racaniello, and you can find me at microbe.tv. I'd like to thank the American Society for Microbiology for their support of TWiM, Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.